Everyone, and now let's talk about inflammation. So here, what I want you guys to actually really to kind of think is in connecting all the things we spoke about the whole innate immune systems and how they work. Coming from the complement, coming from the basically barriers, and also talking about the ability of innate immune cells to actually recognize some sort of you know um, pathogen. So, because once they recognize, then this is what happens, inflammation. So it's literally think about inflammation as the connecting kind of, let's say, theme with all the other stuff that we had. Try to connect all the things you learned here. So what is basically the inflammation is literally a response that is able to recruit effector molecules and phagocytic cells to the site of infection. So what happens in general is something has penetrated into us. For example, be it for you have a wound and where bacteria are now suddenly able to cross from the skin barrier inside to our tissues and blood. And this is extremely dangerous because now we are under attack. So what that means is that we need to start to actually be able to carry out effector molecules and cell delivery to the site of infection to actually come in and kill the microorganisms being infected, but also to induce blood clotting and promote tissue repair because we need, first of all, to stop the bleeding that's happening here and also kind of fix the tissue that has been damaged. And the phenotypic signs of inflammation are four, which is heat, redness, swelling, and pain, which I'm quite sure are literally things they've all have experienced. So I'm quite sure if you guys have ever, I don't know, been playing outside and you've got a splinter, so a small piece of wood has gotten inside your fingers, what you will see is you will actually start to, if you put your hand over there, is that of course now the splinter's inside and now there's literally some inflammation happening. The four things that you will literally feel is heat, you're going to see redness and swelling, and then it's going to be, of course, be painful. Well, all of these four is all because of inflammation, of the response that happens. And we're going to literally, I'm going to tell you why every one of those symptoms comes out. So, as I said, we are technically, when it comes to the inflammatory responses, there's so many things that technically can induce an inflammatory response. They can be non-infectious factors like injury, a burn, or a trauma, chemicals, chemical toxins, and biological drugs. Because what's happening technically is you have damaged cells. You're causing damage. And when you have damage, you have release of DAMPs. These DAMPs are literally kind of seen as a danger or as in some sort of way the immune cells start to actually see this as something is wrong, we need to start going crazy. And of course, there can be infectious factors like viruses, bacteria, and microorganisms. So you start to see, you know, certain PAMPs coming in and activating. So what I'm trying to tell you here is that you need some sort of, let's say, signal to your immune cells that are able to actually recognize, be it either a DAMP or a PAMP. This is then going to start creating your inflammation. So it always starts, as I said, by your innate immune cells, and it's specifically, it's your macrophages. Your macrophages are one of the first cells that are there to actually grab. And what they're going to do is they're literally going to first recognize the harmful stimuli, be it a DAMP or a PAMP, through the PRRs. That's the first thing. Second is the activation of the inflammatory pathways, and this is again through the pathways, the ones that we just spoke about, the NF-kappa B, the MAPK, and this is another pathway that basically I didn't speak about, but which is the JAK-STAT pathway. So these are the three pathways that are mostly will be found in your inflammatory responses because these ones are extremely important in producing inflammatory molecules. If you see There's one that is not there that I spoke about, which is more the basically this IRF3 pathway that is more for viruses. It's not more for the inflammation. So these three signaling pathways are really good at inducing cytokines and chemokines, all these kind of molecules that will induce inflammation, which we'll be talking about shortly. Once those pathways are activated, inflammatory markers are released. As I said, these cytokines and chemokines, and then you have the recruiting of other cells to come in to the site of infection. So this is how and we're going to be talking about literally every single one of them a bit more into details. So as I said, it's literally, in this case, for example, it could be a TLR signaling. It doesn't have to be a TLR signaling. It could be any type of PRR that is literally first recognizing a DAMP or a PAMP, which is then basically in this case either going into the NF-kappa B or the MAPK pathway. This one over here, the IRF3, don't worry about it. This is mostly for viruses. It's mostly these guys and the JAK-STAT pathway. So as we said, you have an immediate alarm phase. This happens literally within seconds to minutes. So as I said, the macrophages are the first ones to detect. They detect the microbes or the DAMs if there's basically some tissue damage, and through the PRRs, they will be able to actually recognize them through the PAMPs or DAMPs if there's tissue damage. This activates this signaling cascade, which then in return starts to release cytokines and chemokines and so many different types of beautiful molecules that are going to create literally inflammation. And let's talk about these different types. So we have cytokines. And the cytokines that are literally released mostly are interleukin 6, TNF alpha, and interleukin 1, interleukin 8, or CXL8. So these are basically the same. So these are cytokines or chemokines that are being released. Interleukin 6, usually it's extremely powerful. It's one of the most, let's say, main cytokines. That what it actually does is it actually activates other immune cells to be activated to be around it. It also increases PRRs in the surrounding immune cells, but it also can have a lot of effects in the vascularization, which I'll be talking shortly. TNF alpha and interleukin 1 are also extremely powerful. They literally can cause fever, actually produce more other cytokines. They can cause chemotaxis, meaning they can bring other immune cells to come in, but they're extremely also important in inducing something called leukocyte adherence and activation of fibroblasts, which we'll be talking about what this means. So all of this is more in the bringing of immune cells to the inflammation part. And then you have interleukin 8 or CXL8. This is a chemokine that's produced by macrophages, which attracts neutrophils to the site. It's now trying to bring them more to the site of infection. But also, what you have, especially when you have tissue damage, this actually, when you have literally tissue damage happening, for example, in this case, for example, here, like blood vessel injuries or damaged vessel walls, you actually have the activation of your clotting system, because now your system is like, okay, damn, there is release of blood. We need to start literally producing blood clots to stop this. And of course, this then activates the kinin system, and it's specifically this bradykinin. And what bradykinin does, it actually causes vasodilation, vascular permeability, and I'll be talking about these a bit more later. But this is what you need to know technically. But also in this kind of case, interleukin 6 actually, it is important because it kind of pushes your liver to produce certain proteins, and specifically your complement system. Actually, your interleukin 6 pushes the liver to produce a lot of complement, and the complement are going to come to the site of infection, and they're literally going to act within seconds, either if there's antibodies there to the classical pathway, or if there's mannose to the mannose binding lectin pathway. So this is usually what it happens. But as of course, as we know, You have the release of C5a, which is also a vasodilator, but also a chemoattractant. It attracts other immune cells to come in. So you of course have opsonization for phagocytosis, but you also have the literally bringing up of the membrane attack complex. And this is all because of interleukin 6 that actually brings them. So this is just mostly in the plasma-derived mediators that that you basically what happens in general, that when you have the blood vessels are literally leaky now or damaged, that you start to have literally the production of blood clots because you need this. But also within other than the macrophages, basically other cells that are also literally immediately there are mast cells and basophils. And mast cells and basophils through the PRRs will also detect PAMPs or DAMPs. And mast cells will release histamine and prostaglandins, which increase literally vascular permeability and also production of other chemokines. Basophils also releases histamine, which also, as I said, increases the permeability and everything. So now, as you can see, I've been saying a lot about vasodilation, about permeability. So all of these literally uh, production of cytokines and chemokines and all the different types of molecules that I spoke about will act upon our vasculature. So what happens is they can actually cause the whole vessels to vasodilate, which basically literally means to open up. When basically the whole um, basically the vessels open up, what happens is you actually have a, a decrease in pressure, and when you decrease pressure, the velocity on how the cells or whatever is in the blood is traveling decreases. And this is important because macrophage immune cells are traveling almost at 200 kilometers an hour. They're going at an extremely high speed. So to kind of help them to reach areas and be able to stop, what you need to do is open up that vessel to decrease pressure and decrease velocity. So you do that by actually releasing all these different types of histamine, prostaglandins, basically this bradykinin, this basically TNF alpha, interleukin 1. These really help a lot, technically, the vasodilation. But also another thing that also helps vasodilation is actually nitric oxide, which is released from macrophages as well. So this is one actually of the vascular phase that actually occurs, and this happens right after the immediate alarm phase. Another thing that we also see is permeability, meaning that you start to see swelling. So by the way, because now this vasodilates, you start to get actually heat because now you're increasing technically the volume, so there's much more blood, so you get actually much more heat and actually redness, but you also get actually something called permeability. You're causing them to actually go into more swelling because you're increasing literally these what do you call it? These uh sorry, I'm just losing a bit because but basically these kind of veins or arteries technically, what you're doing is because they're opening up, you're making them to go actually swell, and they're much more easy to actually permeabilize. So the immune cells that are there are much easier to actually be released. So if you can see here, these are much more constricted. You have like what, three or like let's say five immune cells around there. But once these are actually much more expanded, you help literally these immune cells to actually be able to permeabilize, to leave the either the arteries or the veins to the outside, so you're making them more leaky. So hence, you're basically increasing permeability to go outside. And of course, you with this you get also release of liquids. So hence, this is why you get swelling. And we said all of this is caused by histamines, basically by these leukotrienes that are coming from mast cells, this bradykinin that I spoke about, C3A, C5a from the complement system. These are all causing the vasodilation, which is then leading to edema. But also, what you also see is you're also causing the endothelial cells to be activated, and specifically within your veins or arteries. Because one thing that you want them, so let's imagine you have immune cells that are really growing super fast. Let's imagine they need to stop here. Well, they need to be able to, in some sort of way, stick here, because why do they need to stop here but not way far, like one literally kilometer away, for example? Why here? Well, that's all because in the site of inflammation, what you do is you release certain cytokines or chemokines that I was speaking about, that literally what they do is they upregulate certain glues in that area of the endothelial cells in the veins or the arteries. And these can be, for example, selectins, like for example, B-selectin or E-selectin, or these can be ICAM molecules. P and E-selectins are mostly found in your endothelial, and these are very good, let's say, glues. You can think about them as glues. They really bind to, for example, passing neutrophils that are trying to come in, but they bind to them in a very weak manner. P-selectins, P or E-selectins are very weak. They don't they kind of bind very slowly. And we'll give you good examples and what I mean here. But then you have ICAMs, which are much more stronger. That once they bind to the neutrophil, they can really stop it. The P-selectins, they kind of help it kind of stop. Let's say the neutrophil is going at 200 kilometers per hour. Once it binds to the P-selectin, it can make it drop down to, let's say, 100 kilometers per hour. And then once they're bound to ICAM, they can fully stop to zero. So technically, these ICAMs or these P or E selectins are usually upregulated by certain cytokines or chemokines, for example, through TNF alpha. TNF alpha really upregulates these P selectins or E selectins or these ICAMs. So it's literally the local distribution of TNF alpha that causes these blood vessels to actually become sticky, to release, to actually upregulate these selectins or these ICAM molecules. And of course, neutrophils by tumor necrosis factor, TNF alpha, or literally other, for example, CXL8 or interleukin 8, causes neutrophils to upregulate also their own type of receptors, like these integrins, which are they're basically multiple types. They're basically made of alpha and beta, basically proteins, and it's just different combinations. I don't expect you guys to memorize them. I just want you guys to know that there's integrins in the neutrophils that bind to the P-selectins in the endothelial, or they bind to basically the ICAMs. So it's both the endothelium that releases these glues, but also the neutrophils. And these are mostly activated through cytokines and chemokines. So then basically after now you've literally made your tissue much more, let's say, dilated, you've made it more permeable, and you've made it more sticky. Now you can actually enhance cellular recruitment. So macrophages and the mast cells and the mast cells and the basophils that are there that are releasing all of these kind of chemicals, what they're trying to do is recruit other immune cells to come in. And the recruitment of cells is usually done in four phases. It's the initiation, adhesion and rolling, diaperesis, and migration. So, and usually the first recruited cells are neutrophils. Into the inflammation, as I said, usually the inflammation is mostly macrophages are the ones that are there pushing this, and the first ones that they recruit are neutrophils usually. So let's go into basically the whole process of this. So as I said, the first one is literally the initiation, adhesion, and rolling. So these are the first ones. As I said, these neutrophils that are not trying to be recruited, they're traveling extremely fast. So what happens is, and technically, they first have to be kind of captured. And they usually are captured by the neutrophils that are basically now expressing. So in this case, they're expressing their own prostaglandins. So these are literally their own little beta. So these are basically these integrins that they have over here. So they're literally kind of upregulating them technically. And they're basically the neutrophils are upregulating them because of certain cytokines like TNF alpha, interleukin 1, or interleukin 6. And what happens is that they're able then to also bind to the endothelial cells over here that are expressing, for example, here, their own basically, so this is basically the P or E selectins. So as I said, the PE selectins are weak. They can kind of capture them, but they're still rolling and they're rolling slowly. When are they able to arrest? Is literally when you have the activation of LFA, basically this ICAM. So once you have ICAM, as you can see over here, if so this is the ICAM. Once you have ICAM, this actually stops it. It arrests it. So you always have to think of these selectins, this P and E, that they're literally kind of capturing them. So they're literally causing the initiation, they're causing the adhesion, and they're literally kind of capturing them, and they're still rolling, and then bit by bit they're rolling slowly, slowly, slower until they really are arrested. And the arrested happens through technically these ICAM molecules. So this is what happens. And as I said before, literally TNF alpha, interleukin 1 beta, interleukin 6, they're the ones that are upregulating all these different types of glues in the endothelial cells and in your neutrophils. So once now it's stopped, now it can do diapedesis, which literally means going through the endothelial cells into basically the tissue. And how it's able to do that is either through two ways: paracellular, so in between cells, or transcellular through one cell. And usually it's done through paracellular. It's preferred that way. It doesn't like to be done transcellular in that kind of sense. And of course, to go through inside, let's say, the cell, what happens, of course, as we know in the endothelial cells, there's a lot of tight junctions or adhesion molecules that are literally kind of pushing the endothelial cells together. These are basically in some sort of way broken down or kind of removed to allow this neutrophil to go inside into the area. And then you have migration, and that is basically once it goes to the endothelial cell from basically, so this is literally the inside of a vein or the artery. Now it's in the basically over here. There's a lot of this kind of membrane, this basement membrane, which is a lot of collagen and laminins. They have to be able to actually migrate through it. So this everything that I just talked about is called chemotaxis, which is literally describing the movement of immune cells through basically a gradient. So as I said, in the area of bacteria, basically where the inflammation happening, they're releasing a lot of cytokines and chemokines, and specifically chemokines are the ones that are really attracting the neutrophils. So they're making them come to the area where there's a lot of chemokines in there. And then after that, once you're basically bringing all of these cells, it's time to kill. So the killing can basically, if you attract NK cells, the NK cells start to kill through the release of perforins or granzymes, or then you have basically phagocytes coming up like neutrophils eating up everything that is inside there. So sometimes what happens is that this whole inflammation is extremely strong, and what you actually have is fever because you have so much release of interleukin 1 and interleukin 6 in TNF alpha that they go to the hypothalamus, and what they do is they actually increase this literally kind of COX inhibitor, COX-2, basically enzymes which are literally causing the whole fever production. I'm not expecting you guys to know this, but all I'm trying to expect you guys to know is that in generally interleukin 1, 6, and TNF can really induce fever if this is continuous, if it goes really for a long time. And in some sort of way, you have to have a resolution. You have to actually have some stop signals. And usually, as I was saying before, what happens is that you have a release of certain cytokines and chemokines that are literally there to stop. These could be interleukin 10, which are released by macrophages, or TNF beta. These are literally trying to deactivate all the activated neutrophils or macrophages or NK cells there. It really stops it, and it helps it to actually go into resolution, which is removing the trigger, which is the microbe or debris, to cause the neutrophils to die. It's like, okay, you've been activated too much and we want you to die, and to tell the macrophage to start to clear the dead cells that has been there. But also, most importantly, because as we have said you before, there has been a lot of edema, a lot of swelling because there's been a lot of leakage, is that the lymphatic has to start now drain up the fluid. This is what we mean. So this is really the inflammatory cascade, technically. Thank you so much for listening, and if there's any questions, please let me know.